he can tell us the story. Thank you, Emma.
Yeah, thank you very much, Trevor, for the wonderful introduction and for the invitation. Yes, my name is Ammar Dang Azumi, and I am a postdoctoral researcher in the Biomedical Science Division at KAUST. And today I would be sharing with you our work on SB40 LTAC helicase and how this very interesting helicase melts the origin DNA and unwinds the DNA for replication initiation. I would start with a brief overview on DNA replication and how this process is centered around helicase. And then I will introduce SB40 virus and specifically the LTAC helicase and why it is a suitable model for DNA replication. And then I would go ahead to introduce the set of questions we set to answer and how we were able to show how this helicase can unwind full origin and also the mechanism we propose for general origin unwinding in SB40 virus and also in eukaryotic helicases such as the human CMG. Starting with DNA replication, we know for cell to divide, the DNA needs to be replicated and passed to the progeny in a semi-conservative manner. And this DNA replication is orchestrated by an assembly of enzymes and protein factors called the replisome, which is centered around a helicase. In this illustration, the helicase is shown in as CMG from the two ends. And what this CMG does is it recognizes origin of DNA replication, and then it assembles a double hexameric assembly. And this assembly melts the DNA and then establish two opposing replication forks around the helicase onto which other replication factors are assembled to replicate the genome in a semi-conservative manner. This CMG helicase in human and other eukaryote like yeast is a complex of 6 MCM protein, 2 to 7, that assemble in a ring shape. The ring has a channel that can accommodate DNA, shown here is in yellow. And it has also other loading factors such as CDC45 and the tetrameric genes protein containing PSF1 to 3 and SLD5. Like I said, it's assembled in a ring form, and this ring is a two-tier ring with the end tier forming like a collar and the C tier on the bottom that houses the triple A plus helicase motor, which facilitates hydrolysis of ATP to enable translocation on DNA. And this motor is characterized by a set of conserved Walker A and Walker B motifs, along with the trans-actin arginine finger, which is also conserved. And together, these residues coordinate ATP in the helicase motor and facilitate ATP hydrolysis. I would move. I will switch from CMG to SB40 LTAC and show how it's related to CMG. SB40 virus is called a SMEM40 virus. It's a virus that belongs to the polyoma group of viruses, and it infects monkeys. This virus was first discovered as a monkey kidney culture contaminant during production of poliovirus vaccine. And then it's a double-stranded DNA virus with a circular genome of 5 kilobase pair, and its infection has been detected in various human populations. However, the role of this infection of this virus has been very debatable. It has been linked to several tumors, but the particular implication of the virus with the tumor development is still a subject of active research. This virus encodes a very particular protein called the large tumor antigen, or LTAC, if you like, and it's 708 amino acid protein that has different domains. From the N-terminal, the DNA-J domain that is involved in protein-protein interaction. And then you have the origin binding domain called the OBD, which is the recognition sequence for the origin DNA binding. And also you have the zinc binding domain that supports DNA binding alongside with the OBD. And then at the C terminal, you have the AAA plus ATPase motors that can facilitate ATP hydrolysis. This protein is 82 kilodalton in size as a monomer, and it can hexamerizes to around 500 kilodalton hexameric ring-shaped protein. And like CMG, it's also form a two-tier ring with the N-terminal collar and the C-terminal C-tier ATPase motor. And like in CMG, it also uses the set of conserved Walker A and Walker B motif alongside the transactin arginine finger to facilitate ATP hydrolysis. This protein is the main replicative helicase of the virus, and it specifically recognizes a specific sequence of DNA called the SB40 origin, which is a pseudo-palindromic sequence that is characterized by recognition sequence 4 of them, pen 1 to 4 in blue here highlighted. And onto this sequence is it's onto this sequence that as a large tumor antigen recognizes where to initiate DNA replication. The sequence can be broadly divided into two halves. You have the EP half on the left and the AT half on the right. And in previous years, several crystal structures have been reported of the origin binding domain, this domain in orange, bound to the SB40 origin with the recognition sequence in blue, highlighted here, showing how this origin binding domain interact with the DNA. Also, another crystal structure of the dimer of this protein bound to the EP half of the origin, showing how the hexamer nucleation occurs. And another interesting structure, although very low resolution, was the cryo-EM structure of the double hexameric helicase at the origin that was reported in 2010. However, the DNA was not resolved in the structure, so the mechanism of how this process is going on still was unclear. Going back to the SB40 origin, you have the origin DNA, and this hexameric LTAC recognize the origin through those sequence and assemble a double hexamer like CMG. And then this double hexamer melts the origin and establish two opposing replication forks. And then this is where it gets interesting. SB40 LTAC, SB40 virus does not have replication machinery. So it uses this LTAC to recruit human DNA replication factors such as polymerase alpha, polymerase delta, and pCNA to replicate its own genome. So in a way, the virus uses uses the LTAC protein to hijack human DNA replication factors, and this property of this LTAC and the virus indeed makes it a suitable model to understand a eukaryotic DNA replication process. So then we set out to understand some question on how this SB40 LTAG is operating. Well, we first want to understand how the double hexamer are assembled around the DNA, the SB40 origin, and then we want to see how this double hexameric LTAG unwinds the origin, and we want to also understand how it recruits human DNA replication factors to replicate its own genome. To do this, we start with our first goal to reconstitute the double hexameric helicase with the SB40 origin DNA. So we have an assembly illustrated here, but instead of ATP for hydrolysis, we substitute ATP for a non-hydrolyzable analog, AMP-PNP, such that we trap the complex. And then we froze this sample and acquired around 15,000 cryo-EM movies, which from these movies we were able to classify and fish out the double hexameric assembly, which you can see in the 2D class averages with 2 lobes. And then we were able to reconstruct to have a 3D reconstruction of the complex at 3.7 Armstrong. However, this reconstruction was not suitable for any atomic model building because only one hexamer from the left is ordered, but the OBDs in the middle and the DNA alongside with the second hexamer from the left is very noisy and disordered without any defined density for model building. So I tried to process the data very deeply by splitting the density using signal subtraction. So I split the density of one half of the origin and then classify and recover the density. And also for the second half, I did the same. Still, the resolution of the second half was very poor, 6.1. From this, I made a composite map, and after post-process, you see the density in the middle of the OBD and also the DNA is completely gone. So again, this reconstruction cannot tell us what is going on between the protein and the DNA. So we devised a clever strategy by revisiting the SB40 origin. If you look at the sequence, as I've mentioned before, the sequence can be broadly divided into 2. You have the EP half and the AT half. So we did exactly that. We split the sequence into 2. We have the EP half and the AT half. And then we assemble a single hexamer onto each half. And then if we do this, we think we can recover the information of the double hexamer from the two split halves. So we did exactly that, starting from the first half, the EP half from the left. We assembled a single hexamer on the EP half, and then we trapped it again with a non-hydrolyzable ATP analog. Acquired some cryo-EM movies and classify, and then we could reconstruct a higher resolution EM map at 3.1 Armstrong. And then with this reconstruction, we can identify the DNA from the channel and also from the side view. We can also pinpoint the density of the nucleotide in the helicase motor. And indeed, we could build clearly the model of the DNA alongside the model of the full protein. So what could we understand from this atomic model. So first, what we did, we zoom into the interaction between the protein and the DNA, and what we see is that you have DNA binding loops from the protein that arrange in a staircase conformation around the DNA. And then this staircase conformation was incompatible with double-stranded DNA in the channel. So we have double-stranded DNA from the top until the middle of the channel, and then suddenly we lost density of the second part of the DNA strand. If we zoom even more into the interaction at this channel, we see that on these loops, there are specific histidine residues from all the 6 loops, histidine 5, 1, 3, that intrudes into the DNA minor group and form a hydrogen bond with the nitrogenous basis of the DNA. And this interaction really splits the double-stranded DNA and prevent the Watson-Crick base pairing from occurring. So what we could conclude from this structure is that nucleation of LTAC hexamer triggers the melting of the local melting of DNA in the ATPase channel, even in the absence of ATP hydrolysis. To identify where exactly this melting occurs, because in this structure we do not have base pair resolution. So what we did, we revisited a crystal structure of LTAC dimer bound to the EP half origin, the part that we are currently working with. And that crystal structure has a base pair resolution, has a high resolution that could resolve the base pair. So what we did, we align our structure, our hexamer onto this structure and zoom into the alignment, and we can clearly see that the DNA is perfectly aligned until the region of the melting, and we could really pinpoint the identity of the bases that are melted, which is shown here in this scheme. And also to confirm that this assembly is not, it's a true assembly. This melting is a true melting event, not an artifact of our structural reconstruction. We performed DNA unwinding of this substrate by SB40 LTAC, and we see that melting that occurs in this way is sufficient to open up the DNA and allow the helicase to unwind the DNA, confirming that this is a true melting event, not just an artifact of the reconstruction. So this is the first half. Then now we switch to the second half of the origin, the AT half, and we assemble again a hexamer and trap it with a non-hydrolyzable analog of ATP. We did the same thing, acquire cryo-EM movies of roughly the same size, and average the particles, and again obtain a similar reconstruction at 3.1 Armstrong with clear density of DNA, AMP, PNP, and also this density is sufficient to build atomic model of both the DNA as well as the protein. Again, we try to look at the interaction. We see the same staircase arrangement of the DNA binding loops in the helicase channel. And again, the same set of interactions with histidine 513 in the channel that destabilizes the hydrogen, the Watson-Crick base pairing of the DNA, double-stranded DNA. So we reached the same conclusion from this reconstruction that on the second half of the origin, nucleation of LTAC hexamer is sufficient to trigger melting from both ends. We aligned these two structures from the two halves, and what we found is that they are exactly identical, with with a negligible RMSD of 0.25 Armstrong. So indeed, the same thing is happening from both ends of the origin DNA as the hexameric helicase is assembling. So from here, we begin to build a mechanistic model of how SB40 origin melting occurs. To do this, we revisited the crystal structure of OBD, the origin binding domain, bound to the origin DNA. And then we can see 4 OBDs occupying the 4 pen sequences. But when we align these structures when we align the crystal structure of the dimer from the 2 ends, what we see is that as you start nucleating monomer upon monomer, once the second monomer binds, the OBD cannot reach to its second binding site. So meaning only one OBD can be bound when you start nucleation. Again, when we added onto this alignment the structure of our hexamer from both ends, what we see, even the second OBD that cannot reach the pen sequence has to detach from its position because of steric clash. So what this alignment shows in summary is that once the OBD's function is just for DNA recognition, and once they recognize the DNA, they have to detach to allow nucleation of full hexamer. And then this nucleation is accompanied by melting of origin from both ends of the DNA. And from this kind of analysis, we could pinpoint from both ends where exactly the origin melting is initiated, highlighted here in gray from the 2 ends. And also our composite model was able to fit into the low resolution cryo-EM map of the double hexamer we obtained. So supporting that indeed our strategy to split the 2 halves indeed can recover the information of the double hexamer. To compare this with other eukaryotic helicases like CMG, what we see is starting from left, this is our mechanistic model. What we see is that you have a DNA, double-stranded DNA with DNA binding loops forming a staircase conformation. And the same thing happens if you look at yeast CMG. You have the MCM, the DNA binding loops from MCM forming a staircase arrangement, also accompanied by melting of bases. Again, in a fork structure that is actively translocating CMG, you see the same staircase arrangement. But very interesting observation is that when you have the DNA binding loops in a spiral, in a planar arrangement, not the staircase, then you don't have DNA melting at all. You have intact double-stranded DNA. So this shows that indeed what we showed, that nucleation of the hexamer is what triggers origin melting, not ATP hydrolysis or any special event. Okay, so having established this, we then want to understand how, after this melting, how this helicase translocate on DNA. So Taha Shahid, who was a postdoc with my PI in University of Leicester at the time, set out to understand how this translocation is happening. So what Taha did was he assembled LTAC with a forked DNA. He assembled a single hexamer with a forked DNA in the presence of ATP, but also ADP and other non-hydrolyzable analog. and then obtained a cryo-EM high-resolution cryo-EM reconstruction of the assembly. Then Taha used 3D variability analysis to decompose the motion of this helicase as it translocates and unwind the DNA. And what he found out was summarized in this sort of movie, where you see the DNA binding loops. The one here in cyan is rearranging from its initial position, moving up to capture DNA and pull it down into the channel. So in summary, what Taha showed was that as ATP hydrolysis occurs, the C tearing of the helicase relaxes such that the DNA binding loop can move up and capture DNA at an elevated position and drag it into the channel. And once the DNA is into the channel, the staircase arrangement is reestablished, and that staircase arrangement destabilizes the base pairing, thereby causing melting in the new position of the DNA. We compared the structure of the actively hydrolyzing structure of Taha with my structures, and what we found out is that indeed the DNA binding loop specifically from F subunit moved 10 Armstrong to capture the DNA during translocation. Again, we did the same comparison. If you have this is a crystal structure of LTAC without DNA, showing planar arrangement of the DNA binding loops. And this is Taha's structure with fork that is actively translocating, showing that the DNA binding loops are in the staircase conformation. And the last two are my origin binding structures that we've also shown that they are forming the staircase, the loops are forming staircase conformation around the DNA. And indeed, this staircase conformation is the conformation that triggers origin melting and enables DNA unwinding. And from this, we propose a complete model for melting and unwinding in this cartoon, where melting starts from local melting in the helicase channel from two ends, and then somehow the ATP-driven hydrolysis triggers the sharing of the DNA, and the helicase is decoupled and translocate using this the same staircase arrangement rearrangement during ATP hydrolysis. And together with Taha, we published a joint first author paper in Nature in 2025. So to summarize this, what we have discovered in this work, first we have shown that LTAC binds and melts the origin from both ends. We have also shown the stepwise assembly, the pathway of assembly of the double hexamer from single hexamer. And then most importantly, we have shown that this principle is conserved across eukaryotic helicases, including the human and CMG. And we have shown from Taha's work that we have shown clearly how the mechanism, how the LTAC helicase unwinds DNA during ATP hydrolysis. And this short video summarizes the finding of this work, which I will play now.
Life as we know it begins with a twist, or rather an untwisting. For the first time, KAUST scientists have captured the very first steps of DNA replication when the double helix begins to unwind. Using electron microscopy and AI, the researchers saw how enzymes called helicases use ATP as fuel to break down the DNA's double helix and pull the two strands apart, allowing other enzymes to complete the replication. The study found that as ATP is consumed, it reduces physical constraints that allow the helicase to proceed. Thus, ATP acts as an entropy switch, freeing the helicase to move along the DNA. These new findings not only suggest new approaches for the design of nanomachines, They provide insights into the most fundamental scientific questions about life.
Okay, and with this, I would like to thank my lab members, both present and the past, our collaborators, especially Professor Samir Hamdan and the KAUST Imaging Collabs staff that assist with support during microscopy session, and especially Christos Sava, who is currently in Diamond and who trained me on electron microscopy. And with this, I am happy to take questions. Thank you.
Thank you. Thank you very much, Amar. That was a very, very interesting talk. And for me, it was all completely new. What I found interesting and that I didn't fully understand is what was the basis of the melting? How does the melting actually work?
Okay, interesting question. I would go back to this. Yes, this could work. So what happened is that you have DNA binding loops arranging in a staircase conformation as the hexamer is nucleating. So it starts from a monomer, a dimer, and a trimer. So as you're adding monomers of the protein around the DNA, you are establishing this staircase conformation. But this staircase conformation, as you are establishing this staircase conformation, these histidine residues are competing for hydrogen bond with the complementary strand. So they are competing for establishing hydrogen bond with the nitrogenous basis of one strand. So in a way, competing with the second strand. So this competition is what effectively pushes the other strand away.
So it's the association of the monomers of the hexamer
Yes.
that does the initial breaking.
Yes,
And
exactly.
then the prevention of the reannealing is due to hydrogen bonding with the histidines.
Yes, exactly.
Okay, that's very interesting. Are there any questions that people wanted to ask? There's a question from Sinta which says, does this mean that ATP is not providing the energy for breaking the hydrogen bonds, but for translocation of the hexameric helicase? Please explain the entropic effect.
Yes, so this is exactly what Taha showed in his structure here in this movie. So what happens is that the ATP hydrolysis, because at the interface of this hexamerization, you have the ATP at the interface. So once you have ATP hydrolysis, the interface is broken, so the protein relaxes and the DNA travels down. So the ATP hydrolysis only relaxes the channel such that DNA can travel down, and while DNA is traveling, this staircase arrangement is reestablished and melting occurs. So the ATP hydrolysis, it's like an entropy switch that relaxes the motor and allow DNA to move downward. And as the DNA moves downwards, this staircase arrangement that I've shown earlier, again, compete for the hydrogen bond and melt the DNA. So this is how melting and translocation is coupled.
That's extremely interesting. It's beautiful.
Yeah, that is very interesting. So, yeah. Trevor, there's another question. I don't know if that's probably not you that's with the hand up, but somebody else. But I had a question of my own.
Okay.
Okay, so
I see.
who wants that?
Wolf Dieter, go ahead.
Hi there. I hope my microphone is working.
We can hear you, Wolf Dieter.
Okay, good. Okay. Well, thank you very much for that presentation. So, but does it still apply that the helicase, kind of the hexameric, or that the individual units hydrolyze ATP in a certain sequence, kind of, so that kind of we get this melting in a circular fashion, or is that are you basically saying that kind of that assumption is not correct?
No, it's correct. It's melts. I should have shown the slide, but in the paper, if you read the paper, we have a description of what happened at the interface. So the melting specific, the ATP hydrolysis happens in an order. So in one interface at the top, which we call the BC interface, should be around here. So the ATP hydrolysis happens there. And then this ATP hydrolysis is coupled with the movement of the cyan subunit, movement of the cyan subunit up. So the interface is rearranged, B becomes C, D becomes A. So it goes in a linear fashion. It goes one subunit at one interface at a time. I don't know, I should have shown the slide, but
and then how much translocation happens per ATP, kind of, or how many hydrogen bonds are broken per ATP?
The translocation we estimate is one base pair per cycle.
It's only one base pair. Okay.
It's only one base pair per cycle.
Because I teach this to undergrad students. So that is what I've taught them so far.
Yeah.
And I would just like to know if that was not the case, you actually come to a different conclusion. Okay, but essentially it's still true.
Yes.
Kind of you need one ATP per hydrogen or per base pair, essentially.
Yeah.
Good.
Yes, exactly.
Good. Okay, thank you.
Thanks, Phil, teacher. So was there another question that I missed? Lawrence, did you have your hand up?
I did, and so I wanted to really focus on the melting part, which I think material scientists would probably go, what is meant by melting? Is there an electrostatic repulsion? Is there a conformational change? Is there some kind of trigger that there's a huge rotational change? What constitutes melting?
So basically, melting in the simplest term is just breaking of the Watson-Crick base pairing of the two DNA strands.
Mm-hmm.
So, and like I said, this is from the competition of this histidine residues for hydrogen bond with one of the strands. So instead of forming Watson-Crick base pairing, this histidine competes to form the hydrogen bond and prevent the other strand from bonding.
So this doesn't seem to, this process, do you think it is sort of sequence dependent, or is this a general phenomenon of, I mean, I guess hydrogen bond competitions are kind of a general phenomenon, but
yes,
how much of this is restricted just to this?
I don't think it is, because in this, we have shown, this is our structure from the left. We have shown this staircase and this hydrogen bond pattern that competes and triggers the melting, but also in CMG, in human and yeast CMG, you see this staircase conformation with the MCM DNA binding loops, and also they present an equivalent histidine that can destabilize eukaryotic origin, which is completely different from SB40 origin. Yes, so I don't think it's sequence dependent. Once you have this kind of arrangement and you have this histidine that can compete for the hydrogen bonds, is sufficient to trigger the melting.
I see. And so so if you mutate that histidine to, I don't know what do people substitute histidines for, Trevor?
Well, there isn't an equivalent amino acid.
Right,
but you can put in something hydrophobic and see what happens. But I imagine that would incapacitate the enzyme completely.
Yes, that would be an interesting thing to show.
Yes, I agree. We have not done that, but it's something interesting to check. But I guess the enzyme may not even be functional.
Right. Let me ask another question if there's time and no one else has a question. But there was the point about the recruitment of the replicase.
Yes.
For this virus. how is that recruitment mediated? Is that electrostatic interactions, or is it just that replicators are just always around, and so once you make room, there's room for replicators to fit in?
So actually, this LTAC protein can interact with polymerase alpha in human, and it can also interact with human RPA. So there is even a structure, a crystal structure of the domain of polymerase alpha that's bound to this hexameric helicase. So it's through protein-protein interaction. And once
Right.
it recruits polymerase alpha, then other factors can assemble around.
That's pretty amazing. Sort of the machinery here is interesting and intricate, huh?
Yeah.
Yeah.
There was another question in the chat from somebody that I sent the login to. Just go back again. How do I get? It says, does the L-tag helicase recruit other replication factors in the same manner as the CMG helicase? How? Did you show this?
No, we did not. So CMG is a bit different. First, the recognition of the origin for CMG is not sequence specific as in LTAC. So CMG is loaded by ORC proteins, which are loading factors that loads onto eukaryotic sequences, which eukaryotic origin, which are not sequence specific. So the loading is different. Also, the recruitment of other factors is completely different, because CMG again has loading factors, firing factors like the CDC45 and the genes. And onto this loading firing factors is what other proteins can come and interact. So it's completely different. This is a simplified model and a simplified protein, and this is why we are using it in our lab to understand eukaryotic DNA replication instead of producing 12 or 18 proteins from CMG. This is one protein that can hexamerize and does all the job. So it's different.
It's very interesting.
So there's a question that's not related to the talk. Daisy, do you want to put your question?
Thank you. Can I go ahead, please?
Go ahead.
Okay, thank you. Okay, thank you also to the presenter. So I was asking because I made a presentation during our conference, I think in April, and I haven't been able to get a certificate for it, and I'm asking if anyone knows how I can, because I have emailed the committee, the email that sent me the email address that I got messages from during the conference, and I have got no response. So
Which conference was this, Daisy?
So that was the Biophysics conference in April.
Which biophysics conference?
So it was held on Zoom like this. So that was what it was tagged. I can't tell.
I'll send you a certificate if I can. I'll find your registration and everything.
Oh, thank you so much.
Okay.
So I have a question for you too. Trevor, about the presentation.
Okay, go ahead.
Okay, sorry, I am in geology, so I don't have clear understanding of what you were saying, but I'm trying to relate this to my understanding of maybe crystal growth and stuff like that. So when you put out the structures here, I am trying to ask, could there be any possibility that this growth can be amorphous? And if yes, what could that mean?
That is interesting because first we are not working with crystal, so in a way the hexameric ring pattern is ordered, and indeed a crystal structure of the hexamer has been reported in 2000. So I would say it's more ordered hexameric arrangement that is crystalline than I would say it's amorphous.
Okay. Thank you.
These are very large molecules, and but they're all exactly the same. So these molecules that we're dealing with are little machines where the atoms move in very specific ways to perform their function. So that's the basis of living, of the components of living systems. We have another question from Sinta Yehu. Why can't ATP analogs or mutations alter the entropy switch to allow translocation?
Okay, so I think there is one thing that I forgot to mention is, so for this hexamerization to happen, you need ATP to complete the hexamerization, even though the interfaces are held by protein-protein interaction, but also ATP molecule supports the tight binding of the interfaces. So you need at least a nucleotide triphosphate, monophosphate, or diphosphate to hold the interface. So what ATP hydrolysis does, it relaxes the interface. Why once you hydrolyze ATP, the interface broke and allow the DNA to move into the channel. So if you have ATP analog that cannot be hydrolyzed, then the interface cannot be broken and translocation cannot happen.
Well, Tita, do you have another question?
Yes, I do. You just showed that you have structures with different conformations. Kind of, did you achieve those by using different ATP analogs? Kind of, or how did you get those different stages of the process?
So basically, we know that this is the assembly.
Yeah.
The native assembly is double hexamer, but we try to obtain the double hexamer, which is very poor in resolution. So we split the sequence into two, and we have we trapped one hexamer on each half with the non-hydrolyzable ATP analog. So these are the same state, but from different ends of the sequence. So we want to understand what is going on from with the two hexamers assembled on the origin. But for the different state here, I think you are talking about Taha's
Yes.
structure.
So what was the difference between yours and his? Kind of that you got different.
Yes, so in his own case, the DNA sequence is different. He uses a forked DNA, a DNA that is already opened by design. This is one, and two, he used an active ATP that can be hydrolyzed, such that the protein can translocate. So as the protein is translocating, he decomposed the motion with 3D variability analysis, and then he could reconstruct this movie from that. So this is how we got all the different states of the reaction through substrate and nucleotide manipulation.
Then just going back to the previous slides, you showed that there's a dimer of other molecules in between the two helicases. What is that kind of?
Here?
What is this OBD kind of? What is that kind of?
Yes, so OBD is if you can see, maybe I would show you.
Is that the viral specific protein kind of?
No, no, the OBDs is part of the same protein. So in the first introduction of the protein, you can
Yeah,
see.
sorry, I was a bit late. Yes, so probably
Yes,
missed
yes.
that
So,
part.
so OBD is part of the protein for DNA replication. Yes, so it's part
okay.
of the sequence.
Yeah. So this is part of the whole assembly kind of, and
Yes.
that's keeping the DNA strands apart. Or
Yes, yes, yes. Yeah.
thank you. Very interesting.
Yeah.
Okay. So are there any, there's Jose Maria, welcome.
Thank you.
Do you want to ask your question?
Yes, if possible, yes.
Go ahead.
I have arrived a little bit too late to the conference, but I have seen that you have used 3D for. The matter of the question is related to flexibility, macromolecular flexibility, and tools to study macromolecular flexibility. You
Yes.
have used 3D very clear. You have probably followed the first PCA and going over there. Have you used, I mean, this is the first line of linear methods. There are many deeper approaches. Have you tried any of them to
Yes, so actually during the revision, the reviewers asked for this. So
I was not the reviewer, okay.
yes, so what I am sure we did was we did back projection reconstruction. So we extract particles along the windows of the 3DVA, and we did back projection reconstruction to obtain independent refined maps of the different state we showed.
Yeah, I follow you, but this will, by definition, the first principal component analysis will give you very harmonic and normally relatively large changes. If you want to do anything more detailed, you have to use other methods. What you have done essentially is to check that validity check by
Yes.
using subset of images and checking. This is, of course, something
Yeah.
you should always do.
Yeah.
Okay, thank you.
Yeah, and good to hear from you. I met you in Brazil, I think. 20.
Yeah, I know.
Yeah.
And I have work, as you know, in large T antigen. So
Yeah,
it's
yeah,
it's a DR specimen to me.
yeah, great.
Okay. I think if there are no more questions, I think we'll let Amar return to his activities.
Yeah.
Thank you very much for coming to talk to us, Amar.
Thank you.
I will package the recording and we'll put it on our website.
All right. Thank you
Thank
very
you.
much for having me.
Thank you very much. Thank you.
Thank you.
Thank you.